All right, we're going to go ahead and get started in here. I'm sure people are going to be trickling in as this comes. I have the distinct pleasure of introducing Menno, who's actually joined us all the way from Europe. So he was coming over already and piggybacked this onto his trip. But Menno and I actually met when I was at BD. And you've heard me speak several times about how the fact that we've started looking at hematological malignancies in a more detailed way has brought all of this, not just clarity and innovation to the way that we treat them, but really more predictive precision medicine. And so when I met Menno, I was actually ecstatic because something else that I'd been saying for years is we have to start breaking other indications down into this granularity at the level that we've done for lymphoma and leukemia. So what Menno's here to present to us today is actually these fluorescent protein tetramers that are incredibly innovative, and I hope they leave you inspired. And I really hope to see in the next decade flow cytometry really be applied more and more to allergy. So Menno's background is he has a continuous research grant support in the Netherlands and in Australia. In 2010, he received the Heineken Young Scientist Award from the Royal Netherlands Academy of Science. He's published over 190 papers in international peer-reviewed journals and is named inventor on multiple patent applications. He obtained his PhD from Erasmus University. He held postdoc positions at University of California, San Diego, and Erasmus MC in the Netherlands. Prior to becoming a group leader at Erasmus, he has, since 2015, he has headed the Allergy and Clinical Immunology Laboratory from Monash University, which is where he was when I met him, and Alfred Hospital, where he founded the JMF Research and Diagnostics Center for Immunodeficiency Diseases in Melbourne and was deputy head of department from 2017 to 2023. He was recruited back by Erasmus in 2023 to head the Humoral Immune Memory Laboratory. He's currently responsible for the careers of 14 people with research lines into allergy, primary immune deficiency, and vaccination responses. So we're really in for a treat, and help me welcome him to the stage.
Thank you, Marcia, for the kind introduction, and of course, for having me here. It's been a pleasure, and I'm almost adjusted to the time zone, so bear with me. Pointer is already working. Great. So, as mentioned, my background is really in immunology and mostly B-cell biology. And this is sort of the core scheme of what I work on. And that's really B-cell development. And we've heard a bit about that, especially in malignancy context. But B-cells develop in bone marrow. You have a large pool of B-cells with a unique receptor, so a large repertoire. And only those that encounter antigen, they will actually respond and differentiate into plasma cells that make antibodies. And of course, they're very important for neutralizing potential pathogens, be it bacteria, viruses, in solution. But what's important in this response, you don't just have this antibody response, you also have memory B-cell formation. So these are the same B-cells that have matured, that recognize the same antigen, but instead of becoming an effector cell, they become a resting memory cell. And these are very important because upon a secondary encounter, it's these memory B-cells that respond. And as you can see in the diagram below, which is a classical Janeway picture, this response, this antibody response is much faster, there's no delay, and much greater in magnitude. And that's because these memory cells are much more poised to respond. So they're very important for normal memory, but of course that can also go wrong. So the research that we do is really focused on how these cells are formed, what is their durability, are they really long-lived as we presume they are, and are they plastic? Can we manipulate memory, and especially in the context of disease? And there's several models that we work on, as mentioned, allergy, and I'll focus on that today, but also infection vaccination responses, immunodeficiencies, where often there's a poor antibody response resulting from some upstream problem, often involving B cells. And we did some work on melanoma immunotherapy. So really human model systems, so really patients that are ideally undergoing treatment and understanding what does that treatment do and how can we advance that. Too many buttons. So today I'll focus on allergy, and I think we haven't touched upon that yet, so I'll briefly introduce it. It's really a global health problem. About 30% of people in Western countries have one form of allergy, and it's very diverse. So it really ranges from lung upper respiratory tract, but also, of course, the skin, atopic dermatitis. We have drug allergies that are systemic, food allergies as well, and of course, stinging insects, and they can lead to very severe responses, such as anaphylaxis. And the spectrum is broad because also the causes can be very diverse, be it aero allergens, food allergens, stinging insects, as mentioned, and medication. So dependent, of course, what you're exposed to and how, that indicates what kind of symptoms arise. Despite that being very diverse, the immune response is very similar underlying all of this. And that's what we call the type 1 hypersensitivity response. It's a very acute and strong response triggered by an allergen challenge, and that is directly bound by IgE that is bound to target cells. So this is captured by the high affinity Fc epsilon receptor, and directly upon binding and cross-linking, these cells, mast cells or basophils, directly degranulate, and these effector molecules cause the symptoms. So that's a very quick response, but as you can see, there's IgE involved, and then we know, of course, that's derived from a B cell response, so that has been produced prior. So there has been a sensitization phase where an allergen has been recognized in a type 2 environment rather than a normal environment, and instead of having a classical IgG or IgA response, IgE plasma cells are formed, typically driven by ILC2s and type 2 T cells. And of course, this memory, so the memory B cells, the plasma cells that are there, they lie dormant upon that challenge, and then you see the symptoms. So it's really that memory that you need to target to remove the underlying problem. And we can do that. So the simplest form of treatment is, of course, symptom relief, antihistamines, nasal sprays, or the EpiPen. And nowadays, we also have biologicals becoming available, so anti-IgE, Xolair, or anti-IL-4 receptor alpha, dupilumab. These have direct action, but there's no evidence of any durable effects. So if you take it, no symptoms, the next day, you start over again. But often it is very effective. The only treatment that can really change the course of disease is allergen immunotherapy. And it's actually the oldest form of immunotherapy. The first publication about this is from 1912, and it involves exposure to controlled dose of allergen over time. And this really lengthy process, typically 3 to 5 years of continuous exposure is needed to retrain the immune system. But what really happens is the system is retrained, get a type 1 response or a regulatory T cell response. That's presumed to affect the B cells, driving more of an IgG response and moving away from that IgE response. And that's where it becomes fuzzy. Then you have this desensitization, probably because the antibodies capture away allergen, but there's presumably also other mechanisms that inhibit this degranulation. So we have a very good therapy. It's just very lengthy and unclear in which patients it will work or not. So there's a lot of challenges still to be addressed. And that's, of course, already in the diagnosis, because you need to know which allergen causes disease and ideally which proteins from that allergen. And because it's such a lengthy treatment, we need early biomarkers to know if the treatment will be successful or not. Especially now we're moving on, of course, to biologicals as well, which are very expensive. And underlying all of this is this unknown mechanism of action. We know some changes are happening. We actually don't know what's really causing the effect. That's what we tried to set out, and our approach to this was, when we started on this 10 years ago, was really have a component resolved approach. So really using the allergen specificity to measure, in this case, the sensitization, but also to evaluate immune memory cells and see if we have these biomarkers of treatment success. So we started, of course, with having these specific antigen components, and we set up our own way of making them, really designing recombinant proteins, and we made them recombinant because then we can manipulate what happens. So we really ensured that they have a purification tag, but also a biotinylation tag. So we can put a biotin on the position that we want without affecting the primary structure. And we have different production systems spearheaded at that time by different people in the lab to produce the proteins in the most native kind of way. And using then that biotin that we added to it, we could use strapped evidence, which we can buy commercially, basically in any color of the spectrum, to make tetramers with a fluorescent label to use in flow cytometry. And with this approach, which took us a while to optimize, we presumed we have native folding glycosylation, targeted biotinylation, so no epitope masking. And by having this tetramers, we also improve the avidity and the binding, the strength of binding, the avidity. So just to show you this, and of course we initially set out to do this to detect those B cells that are relevant for disease. So within the large pool of B cells, we have all these specificities, and only a small fraction will be recognizing that specific protein. So to do this, we set it up for multiple different proteins, and we actually make always 2 tetramers of each protein with 2 different fluorochromes, so then we can capture the allergen-specific events on the diagonal. And then, of course, you see that especially with large protein fluorochromes, you see B cells that actually specifically recognize these proteins, especially people working in the lab, I think, because they're exposed to them one way or another. So on the diagonal, we have these specific cells, and we need that because it's literally less than 1%, typically 0.1% of the total fraction has that specificity. This is shown here for bee venom allergen, ryegrass pollen allergen, but we can also do that for influenza, so HA protein, and we did a lot of work on COVID and COVID vaccination. So also for RBDs, we can do that, and the work became even more interesting with all the variants appearing. So within the antigen-specific cells, you can further explore binding to various variant proteins and see what you gain or what you lose over time. A coincidental finding of this was when we initially started to work, you make the proteins, you make a tetramer, you stick it in a tube, and you don't know if it's going to work. And then we thought, in blood, you have basophils. They bind IgE with a high affinity Fc epsilon receptor. So you assume that in a patient on the basophils, there will be IgE specific to that protein. So we thought that could be a nice population to actually test if our proteins actually work. And they did. So if we gate on basophils, you can use this CD123 marker together with IgE because they bind IgE. You can see in a patient on your left that we have a strong fluorescent signal because there is IgE specific for the allergen. So not all the IgE on the basophil, but there's so much that you still have a very strong signal versus an empty strapped evidin, which of course doesn't bind. If you do the same in an unsensitized individual, you don't see any difference between the strapped evidin or the tetramer. So we use this as a positive control, but then we figured this is also a great system to detect allergen sensitization because you can detect it on the target cells and you can choose different fluorochromes. So ideally, you can multiplex that. So we then set out, does that actually work? Can you detect that? And how sensitive specific is that? And how does it compare to what's currently done? So one of the tests that are currently done, especially in a research setting, is the basophil activation test, which basically uses the same principle. So in a test tube, the basophils are stimulated by the allergen, and upon stimulation, they degranulate. They release, of course, effector molecules such as histamine, but several markers that are only bound in the vesicles will then be expressed on the cell surface. And one of these is the tetraspanin CD63. So in unstimulated cells, these are all CD63 negative, but upon stimulation, a large fraction of basophils will become CD63 positive because it's expressed on the surface. So this test is typically done in a dilution series for each individual. And as you can see in controls, in this case for bee venom, there's no signal because they're not activated. And in allergics, at different concentrations of allergen, you see different frequencies of CD63 positivity. And that's why a dilution series is typically done because some react to a very low dose and that signal actually goes down with higher doses and some it's the other way around. So this test works, as you can see, but you really need typically up to 12 to 15 tubes with all the controls to have all the conditions measured. So we did this test in parallel by actually using our tetramers. And instead of looking at one of the markers, we actually look at the fluorescence of our tetramer. And you can see all the cells are positive. So there is, of course, a spread. but typically all the cells have the same amount of allergen-specific IgE. When we do that in the same dilution series, nothing happens in controls, but you nicely see a dose-dependent fluorescent signal on the basophils of patients, which was nice to see because that told us we can actually use that highest concentration. We don't need to do dilution series because the highest concentration is the best discriminative value between patients and controls. So we did this for bee venom. We took unsensitized and sensitized patients. We did the signals and we made ROC curves. And this was a very ideal setting where all the patients had a stronger signal than the controls. So we had 100% sensitivity specificity. We did the same in a cohort of patients with grass pollen allergy. And you can see the same here. And there's a virtual 100% separation of the two, really showing we have a quantitative output, right? In this case, for the ratio of signal over the strep control, very high sensitivity, high specificity. And of course, as I mentioned, there's a potential to multiplex all in one tube. So after these initial studies, these are of course ideal studies with known patients and no controls. We then started to include a large cohort of patients with allergic rhinitis with or without asthma. And these can have different causes. And the cohort we got in Melbourne in Australia, the major causes there are house dust mite, grass pollen, cat and dog allergens. So we put together a tube with backbone markers, so IgE, CD45, CD123, HLA-DR and a viability dye, and then included 8 recombinant proteins on different fluorochromes to measure these sensitization profiles. So we gated the basophils. We used multiple markers for that to really have a clean population to separate them out. And then on this population, we could measure each of the signals. And this is one example. And we took one patient that is really polysensitized to really show you that for all of these components, we get a very clear signal as compared to the control. And what we then did was, of course, separate the cohort in those clinically diagnosed with or without a specific sensitization, and then evaluate the performance of our test on these. So we have our non-sensitized and sensitized, in this case, for house dust mite. And for house dust mite, we use 2 major allergens, DERP1 and DERP2. And you can see the areas under the curve become a bit lower now because we're really looking at a patient population and not stratified patients controls, but still very high areas under the curve. And what's important here, we of course have a couple of patients that are negative for either of the signals. So the question then is, are these false negatives? Or are these patients just not sensitized to the one, but sensitized to the other protein? So if we do a 2D plot analysis, because we do flow cytometry, we like to do 2D plots, you can clearly see that most of the ones that are negative for DERP2 are actually positive for DERP1 and vice versa. So we still get a very high sensitivity and specificity if we use these 2 components in a single assay. So we did the same for grass pollen. We also had 2 components in the tube, LOLP1 and LOLP5. We also see some come below the cutoff. Areas on the curve are still very high. They're well above 0.9. And when we do the double discrimination, we get even higher sensitivity, specificity for this test to discriminate. So these were the most successful. For cat and dog, we only included 1 allergen. And of course, in hindsight, that's always a mistake. We see lower areas on the curve. There's lower sensitivity. And so we see a lot of patients that actually don't have a positive signal in this assay. But when we then looked into the literature, it's very clear that much lower percentages of patients are sensitized to this single molecule. So for cat, a very likely other candidates to include would be FeLD4 or FeLD7. And for dog, it's even lower for CanF1. So it's very important, very likely important to include CanF5 or maybe CanF3 to increase that sensitivity. So we think this is more of a biological limitation than a technical limitation. So to conclude the first part on the basophils, so we can show you now we can have component resolved differential diagnosis by using flow cytometry assay. very high sensitivity and specificity already with 2 components. And of course, there's potential to increase the number. We can make it adaptable for regional variation. I did not show you, but I used ryegrass because that's the common grass species in Melbourne. In Europe, you would use timothy, which is related but slightly different, and you can exchange the allergens. And what we're now actually going for is food allergies. So we do a large study in the Netherlands with the pediatrics department to have a similar sort of differential diagnosis of the major foods: peanut, tree nuts, milk, hen's egg, and wheat. And we're looking now also into drug allergies because for medication allergies, the serology tests are very poor. So there's really a need for a new diagnostic test. So we're leaning into that. So that's really for the diagnosis and differential diagnosis. The second part, I will focus on biomarkers of treatment. And there's a lot of literature, of course, on biomarkers. There's different definitions, and I just put one up, which is very lengthy. But very important is, of course, it should be easily measured, readily available, and hence why we often use blood. High sensitivity, specificity, as I mentioned. Ideally, it's quantitative, or it's quick. And of course, it's also quantitative, so you can measure the effective response. And you want to have a prognostic value, so you measure it early, and you know for a late outcome. And optimally, it will be biologically possible. So the marker is not just a correlation, but it's actually really involved in the process. So with that in mind, we studied patients, and I've shown you all the patients before treatment, but both for the bee venom and for the grass pollen allergies, a lot of these patients were treated by immunotherapy. And for grass pollen, this was what we call sublingual immunotherapy. So that's a daily tablet that's put under the tongue. And in Melbourne, that's typically done 4 months before the pollen season, because during the pollen season, the counts are very consistently high. So it's thought not to overdo it and have both exposure and the treatment. So we have this 4-month treatment, and we measured patients before and after these 4 months. And what we actually did was sort those allergen-specific B cells, and we did what any biologist would do when they don't know what they're going to find. We did single-cell RNA sequencing on these single cells. And of course, these are very rare events, so we had very low cell numbers. On the other hand, we had more than 500 unique events to do measurements with. And took us a while to do the dimensionality reduction and get a good measure, because these are all memory B cells. They're very similar to each other. So any t-SNE or other plot that we made looked like a nice blob, but nothing concrete came out of it until we used this pseudo-time analysis. And that's really, it infers a pattern of differentiation, which would make sense because we would expect that the cells after treatment would be further exposed and further differentiated. And we got this sort of figure. And what we found is that it seemed to make sense because most of the cells on the left would be IgM positive, and most of these in the later 2 clusters here would be class switched. So it seemed to have this pattern. And when we then separated the cells from before treatment versus after treatment, we don't see a clear distinction of 2 populations, but we do see a shift that before treatment, these first 2 clusters are the most dominant ones, whereas after treatment, these clusters 3 and 4 are expanded. So we see a shift in the composition of the compartment rather than really changing from A to B. And when we look at markers that are uniquely different between these early clusters and the late ones, we found multiple, of course, and we focused on surface markers because we could then validate them by flow cytometry. So we had 14 surface markers. We tested 3 of them. In the end, we should have tested the 4th one, of course. So this was integrin beta-1, CD29, Fc epsilon receptor 2, CD23, and IL-13 receptor alpha. And we validated that with flow cytometry, and we could show, especially for the CD29 and CD23, that before versus after treatment, more cells express these markers. So this is just an illustration of a single patient, but we do this paired before and after treatment. So this is on the ryegrass pollen allergy cohort. In blue, we have patients that did not undergo treatment, so 2 time points, and in red, they underwent treatment between the 2 time points. And you can see that only on treatment for the CD29 and the CD23, expression went up. IL-13 receptor did not go up, but I'll show you later. IL-4 receptor, very related, did go up and was more significant on flow cytometry. And when we separate that difference, especially for CD29, we could really nicely separate the patients that had treatment versus patients that did not have treatment. So this was all done for ryegrass pollen. Similarly, we had patients treated for bee venom allergy with a subcutaneous immunotherapy, and that's a 2-month interval. And we see the same pattern of CD29 and CD23 going up. So it's not just one allergen or one form of treatment. It really seems to be the same, irrespective of the initial trigger or the way the immunotherapy is given. So of course, the question then is CD29, an integrin molecule, why would that go up and would it have any function, which we didn't realize would have either. And that's really, is it biologically plausible? And just before we had this data, a paper came out in JXMed in a mouse model where they took out CD29 specifically on B cells, and they actually found with different ways of measurements that if you stimulate the B cell receptor, the signals actually were higher, be it calcium flux, phospho-Sig or phospholin. So it seemed that in absence of CD29, the B cell receptors signaled stronger. So our hypothesis is now that by increasing CD29 levels on the surface, the B cells are actually less responsive, so less responsive to differentiate. And that could be one way to sort of turn off these cells already early on treatment. So that's what I said. And of course, that's early on treatment, and we found those markers. And then soon after we published these markers, these 2 papers came out actually using the same markers that we found to actually identify this different type of memory B cell population, which they typed a type 2 B cell population, which is separate from the classical memory. And as I mentioned, these are the same markers, the IL-13, the IL-4 receptor, and the CD23, which we also found upregulated after treatment. But of course, the proposition there was, and they showed it very nicely, that this is actually the pathogenic population. So this subset of cells, they express IgG, but they have very high level of IgE germline transcript. So upon stimulation, they switch to IgE, and they actually drive that IgE response. So that's the pathogenic population in allergy. And we were a bit confused because, of course, we saw after treatment, this presumed to be pathogenic population was expanded, even though the patients on treatment did very well. So the question was indeed, are the cells that we see these type 2 memory B cells, and do they expand, and what does that actually mean? So we then reevaluated our data. So instead of looking at these as markers on our cells, we actually use these markers to define the population. So we have our type 2 memory B cells defined as CD23 positive. And we can do that in total B cells, but also in the allergen-specific B cells. So this is for LOLP1, so for ryegrass pollen. And you already see that the picture looks very different if you look antigen specifically, and the same with the bee venom. And if you then evaluate the fraction of these type 2 memory B cells within the total population, that's around 5 to 10%. But with the allergen-specific populations in these patients, a much larger fraction has this type 2 phenotype. So it is really associated with the allergy, really assuming that that's the pathogenic population. But what then happens with treatment? So we, of course, had these patients also after treatment, either for ryegrass pollen or for bee venom. On the left here is the untreated patients for grass pollen. On the right, the treated patients. And we have the bee venom. So these are the total memory B cells. And as you can see, not much actually happens between the 2 time points. But if we then evaluate the allergen-specific cells, only in the treatment groups, we see that this population expands after treatment. So really confirming that we can find these type 2 memory B cells. They really seem to be associated with allergy, but they expand. So what does that mean? And are they still pathogenic after treatment? So of course, then we evaluated their phenotype further. We know upon treatment with immunotherapy, patients make IgG4, and that's typically associated with treatment reliance. So it's really induced by treatment. And we see that in serum, both for ryegrass pollen and bee venom upon treatment. When we then evaluate, of course, the cells, I've shown you this data, but when we look on those type 2 memory B cells, how many of them actually express IgG4, we also see that that goes up. So we do see that these type 2 memory B cells expand, but they change, and actually more of these express IgG4. And also more of these, it's a bit wild, but we do see in the paired analysis it goes up, more of them express CD29. So it really seems that these cells expand, very likely because they get triggered by allergen exposure. But of course, what changes then is this upregulation of these markers that dampen their response. And we think that's the early response. And the question, of course, is then what happens later during immunotherapy. So after we did all this, we started a new study, and this was for house dust mite. And now we followed patients for 18 months on treatment with daily tablets under the tongue. And we followed up for several time points, up to 18 months. And we used also sublingual immunotherapy, the same as for the grass pollen allergy before. So we measured various clinical markers, so how the patients did on treatment. And as you can see, the visual analog score, so that's self-reported disease symptoms. We already see that that goes down at 4 months and stays much significantly lower than pre-treatment. And we see also the medication score, so that's the use of immediate relief, so antihistamines or others. Also that intake was lower. So it's not just a subjective score, but also objectively they did not need as much other symptom relief as before treatment. We evaluated the serology, and what we typically see in these cohorts is that initially IgE levels go up and then slowly go down, and we see that both against DERPI1 and DERPI2, so the two major allergens. And we see that over time there's a gradual increase in IgG4. So it's clear that both clinically and immunologically, the treatment is doing something with the patients. So that's a perfect situation for us to then evaluate the memory B cells. So we did this the same as reported before. We evaluated the memory B cells and especially this type 2 population that's positive for the IL-4 receptor and CD23. So within total memory B cells, we don't see any change over time of this type 2 population, even though these patients are treated daily. But when we then evaluate allergen-specific cells, either for DERPI1 or for DERPI2, we then evaluate the percentage of type 2 memory B cells. Of course, these start already higher, as we've seen for bee venom and grass pollen. But we see initially, after 4 and 12 months, these levels go up, similar to what we've seen for the other two allergies. But what is interesting now is that after 12 months, or at 18 months, these numbers start to decline again. So it seems that following this initial expansion with upregulation of CD29, long-term treatment starts to remove this phenotype. And what we're now evaluating is really these patients up to 36 months, because that's what's typically found to be effective for durable unresponsiveness, to see if we can really get rid of these cells completely. And if actually having these cells or losing these cells is a prognostic marker of durable treatment effects. So that's where we're at for our biomarkers. So we've shown indeed that the biomarkers that we found are indicative of this type 2 phenotype. These cells are prone to switch to IgE and are important for the allergic sensitization in patients. We see that these cells early on treatment change their phenotype, and that probably already causes that desensitization during treatment, but that you need to remove this type 2 memory B cell subset before you can really discontinue treatment and have what we call then the sustained unresponsiveness. So that's what we're looking into now. And of course, what we find very important now is to see if we can develop these sort of assays for clinical assays, both for differential diagnosis, but also for treatment monitoring. So finally, I'd like to thank all the people involved. Most of the work I presented was done in Melbourne at Monash University with the team there. And especially Craig started a lot of the work. And Kathy took that over with support in the lab, and our clinical colleagues, Professor Robin O'Hare and Mark Hugh, who've of course collaborated really well, and through them we had access to the patients on treatment. Happy to take any questions. Thank you.
So I'll start the question out. I might have missed it in your talk, but really one of the things you made me think of is like rituximab and how that anti-CD20 was used to deplete B cells, and how we learned through CLL and other treatments that it needs to go all the way to depletion, and then you need to monitor its return. And actually for autoimmunity, we learned that you could predict relapse 3 months before it would happen, if you could actually find CD19, even as simple as that. So is that something that you see this test being able to be done with? And what is the drug that depletes that particular 29 positive population? Is that the marker?
Yeah, so well, we think, in this case, of course, you treat with the allergen.
Okay.
So you really only trigger the cells that actually recognize the allergen. What we think is that you need to get rid of the cells. And that doesn't happen in the first year. You actually expand them. It doesn't happen at 18 months. So it takes probably at least 2 to 3 years. And we're looking at that now. And probably we need to define a threshold under which we can say if we stop now, you'll still have durable effects. But it will take some time to find out. Mm-hmm. Alternatives would be, of course, using biologicals. I don't think you'll start rituximab on allergies.
No.
But what is now currently being used is dupilumab, which seems to work very well, especially for atopic dermatitis, but also other indications. And that, of course, targets IL-4 receptor alpha, which is one of the markers that we find on the type 2 memory B cells. Yeah. So be very keen to evaluate that, because then you don't take out all the B cells, but only the type 2. And if you can go deep enough to really get rid of them. Yeah.
Thank you. Other questions from the audience?
Very nice talk. Thank you. I was curious, what's the longest duration that patients have been on like the sublingual tablet or something like that? And do you have any data beyond, the 18 months you showed?
No. So typically the recommendation in the clinic is 3 to 5 years. And often, they take it as long as possible. But the difficulty with that is compliance. Therapy compliance, which is often very low. And that's why a large fraction of patients actually, it doesn't work, because probably they're not compliant enough. Because we do research studies, typically all the patients are very compliant. So we can't really compare responders versus non-responders yet. And all the clinical studies go for 1 year, because it's just too expensive to do it any longer. So the fact that we now already start to have data from 3 years is very unique. It's a small cohort, but it's the only data out there. So what we now try to do is turn it around and see if we can include patients that are actually into their 3rd year of treatment, and actually start measuring from that time point onwards, and then looking at discontinuation, what happens then. Yeah.
Thank you.
What other applications do you see these tetramers? Because you mentioned a little bit that you were looking at flu and had a pretty fun role in COVID. What else can they be applied to, and how did you think to use them? Like, where'd the idea come from, and where else can it be applied?
Well, we're not the only ones, of course. And some other groups have used them, often just getting proteins off the shelf, biotinylate them and use them, which works in some cases, but often...
Yeah, they have a bad rap.
I'm very happy with some of the flow plots I see in the literature.
Yep.
And that's why we took longer to really make our own antigens and have these strong signals. I do think it's very important for measuring vaccine response and actually showing what you get upon vaccination. But we're now looking into, with colleagues in Rotterdam, is also in autoimmune diseases. Can we find these pathogenic cells responding to autoantigens or to disease-related antigens, and then actually monitor those cells with all the treatments that are now available. Yeah.
Yep. That's what I wanted him to get to. So these have a bad rap because they weren't maybe applied as, and this goes back to something I said, maybe not when I introduced him, but to someone else. So he had worked with the Euroflow group, right? And I'm not saying that that's the whole thing, but you kind of looked at what does it look like to approach something in a very standardized way and end up with a data set like this. Because a lot of what we learned about rituximab came from a 13-year data set on CLL. So the fact that you're building that information is crucial.
Yeah.
Yeah. So thank you.
No worries. Thank you.